Hello students, my name is Niyati Said and thanks for watching Edupedia word videos. My topic for the presentation is the 10th section of the chapter Morphology of Flowering Plants students. In this section of the video, we will discuss about the anatomy of monocot root. Okay. So let's proceed towards our topic, that is anatomy of monocot root. Anatomy means internal morphology. Okay, so we will study about the internal morphology of monocot root. Okay, so as I told you, for a better understanding of tissue organization of roots, be it roots, stem or leaves, it is convenient to study the transverse section of that particular organ. Okay. So this is the transverse section of the monocot root. Okay. The anatomy of the monocot root is very much similar to the dicot root in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundles and pith. As compared to the dicot roots, which have fewer xylem bundles, there are usually more than 6 xylem bundles in the monocot root. And please note that pith is large and well developed in case of monocotyledonous root. Okay, so this is the transverse section of monocot root students. Okay, let us study the anatomical feature of monocot roots from out to in, as we have studied in dicot root, that is from out to in. Okay, so what is this? This is epiblemma, as I have taught you earlier in dicot roots, and it contains long unicellular hairs. Okay, so that's why it is also called as piliferous layer. Students, don't get confused with epidermis and epiblemma. Epidermis, epiblemma and piliferous layer, they both are same, that is the outermost layer. Okay, it is also called as piliferous layer, as I just told you. Okay, and these are the root hairs, student. Okay, so you can say that epiblemma, it is the outermost covering of the root formed by a single layer of compactly arranged barrel-shaped parenchyma cells. The cells, they are characteristically thin-walled, as you can see, because they are parenchymatous in nature. The cell wall is very thin in nature. So these cells, they have characteristically thin-walled since they are involved in absorption of water. A cuticle and stomata are absent. Some of the epiblemma cells, they are produced into a long unicellular projections, as you can see. And these projections, they are called as root hair. Hence, epiblemma is known as piliferous layer, students. Okay, so don't get confused with the epiblemma and the piliferous and epidermis. Okay, they both are same. Okay, rather all three are same, that is epiblemma, epidermis and piliferous layer. Okay, now second layer is cortex. Students, cortex is the major component of a ground tissue of the root. It is represented by several layers of loosely arranged parenchyma cells. Intercellular spaces are prominent, as you can see that this is the intercellular space. Can you see the intercellular space present in the epiblemma? No. There's no intercellular space present between the parenchyma cells of the epiblemma. But in cortex, these cells, they are very loosely adhered with each other. That's the reason intercellular space is very pronounced. Okay? Students, The cortex is mainly meant for storage of water and food too. Okay, the cells, they also allow a free movement of water into the xylem vessels. Okay, and this red layer is exodermis. Okay, cortical cells, they store food, starch and water. Okay, please note that. But this cortex, students, these cells, they allow a free movement of water into the xylem vessels. So that's why the cortex is mainly meant for storage of waters. Okay. Now comes endodermis, students. This is endodermis. Okay. It is the innermost layer of cortex formed by compactly arranged barrel-shaped cells. Students, this black highlighted is barrel-shaped cells. Some of the cells in the endodermis, they are thin-walled and they are known as passage cells. The passage cells, they allow water to pass into the xylem vessel. The remaining cells in the endodermis, they are characterized by the presence of thickening on their radial wall, and these thickenings, they are called as Casparian thickenings or Casparian band, students. Okay. So this is what it is. The one which is highlighted with the green color, that is your barrel-shaped cells. Okay. The radial wall, it is made of a Casparian strips. It is just similar with the Dicot root. Okay. There also it was barrel-shaped cells, and the radial walls, which is subinized, that is deposition of suberin, it forms Casparian strips. Okay, they are formed by the deposition of a waxy substance which is called as suberin, students. Okay, the Casparian thickening, they play an important role in creating and maintaining a physical force called root pressure. Okay, so you can say that Casparian band, it plays an important role in creating and maintaining a physical force which is called as root pressure, students. Okay, so I hope it is clear to all of you now that we have studied the internal anatomy from out to in. That means the outer layer was the epiblemma, then exodermis, cortex, endodermis. Okay, so what we have studied till now, that this is the epiblemma. It is the outermost covering of the root, which is formed by a single layer of compactly arranged barrel-shaped parenchyma cells. The cells, they are characterized thin-walled since they are involved in absorption of water. A cuticle and stomata is absent. And as you can see that some of the epiblemma, not all, students, okay, can you see all cells of a epiblemma bearing root hairs? No. So some of the epiblemma cells, they are produced into a long unicellular projections, which we call it as root hairs, students. Okay, that's why epiblemma is also known as piliferous layer, students. Then comes cortex. Leave it exodermis. Come to the cortex. Cortex is the major component of the ground tissue of root. It is represented by several layers of loosely arranged parenchyma cells, and intercellular spaces are prominent. The cortex is mainly meant for storage of water. The cells, they also allow a free movement of water into the xylem vessels, student. Now come to the endodermis. As I told you that it is the innermost layer of the cortex. See, this is the cortex and this is the inner layer of the cortex, which is formed by compactly arranged barrel-shaped cells. See, these are barrel-shaped cells and there is no intercellular spaces present. Can you see the intercellular spaces present between the barrel-shaped cells? No. Okay. And these passage cells, they allow water to pass into xylem vessel, and the remaining cells in the endodermis, they are characterized by the presence of thickening on their radial wall, which is known as Casparian thickness. Okay, and it plays an important role in creating and maintaining a physical force which is called as root pressure, through which ascent of sap takes place. So these are the facet cells, students. Okay. Now, as I told you that epiblemma, it is the outermost covering of the root formed from a single layer of compactly arranged barrel-shaped parenchyma cells. Okay. The cells, they are characteristically thin-walled since they are involved in absorption of water. That's why epiblemma is also known as piliferous layer, because they are involved in absorption of water. Main role is absorption of water. A cuticle and stomata, they are absent, as I told you. And some of the epiblemma cells, students, not all epiblemma cells bear unicellular projections, which is called as root hairs. Only some of the epiblemma cells, they are produced into a long unicellular projections, which is called as root hair. Hence, epiblemma is also known as piliferous layer, students. Okay. Now comes cortex, which is major component of the ground tissue of the root. As you can see, this is the epiblemma. These are the root hairs or the projections. Now this is the major portion which we call it as cortex. It is represented by several layers of the loosely adhered parenchyma cells with each other. You can see the intercellular spaces because they are loosely adhered, and the cortex is mainly meant for storage of water. Okay, the cells, they also allow a free movement of water into the xylem vessels. Now where is xylem? This is xylem, students. This is phloem, this is xylem, this is phloem, this is xylem, and this is the connective tissue, or it is also known as connective or conjunctive tissue. Okay, students, this is metaxylem, the bigger one, and the smaller one is your protoxylem, and this layer, green one, is endodermis, and beneath the green layer there is pericycle. Okay, and where is pith? Pith is here. This is pith portion. Okay, and these are the Casparian strips. That means it is formed due to the suberin, that is waxy substance. Okay, now comes the innermost layer, which is endodermis. Okay, so this is your endodermis, this one. Okay, so it is the innermost layer of the cortex, which is formed by compactly arranged barrel-shaped cells. Some of the cells in the endodermis, they are thin-walled and they are known as facet cells, students. The facet cells, they allow water to pass into the xylem vessel, as I told you. And the remaining cells in the endodermis, they are characterized by the presence of thickening on their radial wall, and that is known as Casparian thickening. Okay, and they are formed by the deposition of suberin, which is a waxy substance. And the Casparian thickening, they play an important role in creating and maintaining a physical force, which is called as root pressure, students. Okay, now comes stele. Okay, pericycle and conjunctive tissue. Students, stele is the central cylinder of the root, consisting of pericycle, okay, conjunctive tissue, pith, and vascular bundle. Okay, so stele is the central cylinder of the root, consisting of pericycle, conjunctive tissue, pith, and vascular bundle. Okay, now comes pericycle. Pericycle is the outermost covering of the stele. Okay, so this one is your pericycle, which is represented by a single layer of parenchyma cell, students. Okay, now come to the conjunctive tissue. Conjunctive tissue is this. It is represented by loosely arranged parenchyma cells, which is found between vascular bundles. And what are vascular bundles? Xylem and phloem. So this is xylem, this is phloem. So conjunctive tissue, it is a connecting link that separates phloem from xylem. Okay, and they are loosely arranged parenchyma cells, which is found in between the vascular bundles. And the cells, they are specialized for storage of water students. Okay. Now part 2. This is how it is. That means endodermis, pericycle, stele, conjunctive tissue, xylem and phloem. Okay. So this is the single layer of parenchymatous cell which is known as pericycle. Pericycle, it surrounds the stele as I told you. Okay. Stele is composed of pericycle, metaxylem, protoxylem. Students, please note that protoxylem here in this is situated outside and the metaxylem, it is situated inside. Okay. Xylem is exact. That means when protoxylem is present outside and metaxylem is present inside, then that is known as exact. So the xylem in monocot root is exact, just like dicot root. Okay. Recollect that roots show radial vascular bundle. Okay. So here it is phloem. So it is forming an alternate fashion of xylem, phloem, xylem, phloem. See, this is phloem, this is xylem, phloem, xylem, phloem, xylem, phloem, xylem. So it is forming an alternate fashion. Okay, and thus the roots, they have radial vascular bundles, right? Now this is your phloem, right? The rest of the cells around the vascular bundle are parenchymatous. Okay. So this is parenchymatous cells. Okay. These are parenchymatous cells. These are connective tissues which separates xylem from phloem and phloem from xylem. Okay. Because xylem and phloem, they are forming an alternate patch. Like first there is xylem, then phloem, xylem, then phloem. Okay. So that's how it is. It is also called as conductive tissue, which is found between xylem and phloem. As I told you that it separates xylem from phloem. That means it separates from one vascular bundle to another vascular bundle. Okay. And this is pith. Central part of the stele is large and developed. Okay. Now come to the TS of the monocot root. Okay. So this is your epiblema, which is also known as piliferous layer, because it has long unicellular protrudings which is called as root hairs. Okay. So this is your epiblema with root hair. Okay. Now this is your exodermis. This is your cortex. This is your endodermis. Okay. These are the suberized cells. Okay. These are the passage cells. And what is this? This is pericycle. This is protoxylem. Okay. This is metaxylem. This is phloem. And these are the connective tissue. And this is pith. Okay. So what is pith? Pith is this portion. It is the innermost region of the root representing the central axis. Okay. It is composed of few loosely arranged parenchyma cells. Now comes vascular bundles. Okay. They are radial in arrangement because I just taught you that it contains an alternate fashion xylem and phloem. So this is xylem, phloem, xylem, phloem, xylem, phloem, xylem, phloem, xylem, phloem. Okay. So that's how it is, and hence the condition is described as polyarch. Okay. And xylem is described as exact. Okay. Then comes your connective tissue. These green tissues are your connective tissue, which is represented by loosely arranged parenchyma cells, which is found in between the vascular bundles, and they are specialized for storage of water students. Okay. Then comes your pericycle. This is your pericycle. This red one is your pericycle and the brown one is your endodermis. So first we will focus on the pericycle. This pericycle is the outermost covering of the stele, represented by single most layer of parenchyma cell, students. And what is a stele? Stele is a central cylinder of the root consisting of pericycle, which is this red one, then conjunctive tissue, which is green one, pith, which is the central portion, and the vascular bundle, which is known as xylem, phloem. So this all constitutes and forms stele. Okay, then above it is endodermis, which is the innermost layer of the cortex, which is formed by compactly arranged barrel-shaped cells, students. And some of the cells in the endodermis, they are thin-walled and they are known as facet cells. The facet cells, they allow water to pass into the xylem vessel, and the remaining cells in the endodermis, they are characterized by presence of thickenings on their radial walls, and that thickenings are known as Casparian thickenings, students. Okay, then comes your cortex, which forms the major part of the ground tissue of the root, students. It is represented by several layers of loosely arranged parenchymatous cells. And as you can see that intercellular spaces are prominent, as you can see, because they are very loosely adhered with each other, right? So the intercellular spaces, they are very much prominent. And the cortex is mainly meant for storage of water. The cells, they allow a free movement of water into the xylem vessel. Okay. Then comes your epiblemma, which is the outermost covering of the root, which is formed by single layer of compactly arranged barrel-shaped parenchyma cells. And these cells, they are characteristically thin-walled, right? And they are involved in absorption of water. Some of the epiblemma cells, they are produced into long unicellular protrusions, which is called as root hair, and thus epiblemma is also known as piliferous layer, students. Okay. And please note that in monocot root, pith is larger. And number of the group of xylem and phloem is more than 6. That's why it is called as polyarch. See, 1, 2, 3, 4, 5, 6, 7, 8. Okay, more than 6. That's why it is called as polyarch. Okay. 1, 2, 3, 4, 5, 6, 7, 8. Right? So that's why it is called as polyarch, students. Okay. So the stele is polyarch. Now, what's the anatomical difference between the two types of roots, students? So this is your Dicot root transfer section. And what is this? This is the TS of the monocot root, students. Okay. So what's the anatomical difference? See, 1, 2, 3 and 4. Hence, stele is diarch to hexarch. Okay. So in Dicot root, we have studied that stele could be from diarch to hexarch. Diarch means 2, hexarch means 6. Okay. So 1, 2, 3 and 4. Because it is 4, hence it is called as tetrarch, which comes in the range of diarch to exarch. Okay. Now 1, 2, 3, 4, 5, 6, 7, 8. Hence, stele is polyarch, that is more than 6, students. Okay. See, connective tissue, they are parenchyma in nature. Connective tissue could be parenchyma and sclerenchyma in case of monocot roots. That's the difference. Okay. That's the difference you can find between these two. Okay. And the conjunctive tissue in case of monocot root is sclerenchymatous. So it does not produce vascular cambium. Okay. So no secondary growth is seen. Then vascular cambium is there, but vascular cambium is not here. See, this is the pith, which is very small, but it has a pith which is larger. Okay. So this is the pith which we see is larger one in case of monocot root, students. So what is the difference between monocot root and dicot root? So this is the monocot root, in which pericycle produces only lateral roots. Number of the xylem and the phloem are numerous, that is around 8 to many. Xylem vessels, they are over or rounded. Conjunctive tissue are mostly sclerenchymatous, but sometimes it could be parenchymatous too. Pith is large, as you can see, and it is very well developed in case of monocot root, and no secondary growth is seen in it. The conjunctive tissue is sclerenchymatous, so it does not produce vascular cambium. For example, colocasia, musa and maize. These are the 3 examples of the monocot root, students. Now comes your dicot root. Pericycle, it gives rise to lateral roots, cambium and the part of vascular cambium, okay, in case of dicot root. And the number of xylem and phloem are limited from 2 to 6, that is from diarch to hexarch. And xylem vessels are angular or polygonal in transverse section, okay, in case of dicot root. And conjunctive tissues are parenchymatous in nature. Pith is absent or very small in case of dicot root, students. And thus, secondary growth occurs, okay, because the parenchymatous conjunctive tissue, which you are seeing these green one, they are parenchymatous conjunctive tissue, they form vascular cambium. For example, it is seen in Cicer, Ficus, Gram, P and Limanthemum. Okay, so these were the anatomical difference between dicot root and monocot root. Now let's see the diagnostic features of monocot stem. That's how you can identify like it is monocot root or stem. Okay, so this was all about the morphology of flowering plant section, that is anatomy of monocot root. So in my next section of the presentation, we will study about the monocot stem and its diagnostic features. Okay, so till then, stay tuned and keep watching Edupedia word videos. Thank you.